Welcome back to The Deep Dive, the place where we take stacks of complicated scientific sources, from research articles to comprehensive biochemistry texts, and turn them into the essential knowledge you need, delivered with surprising facts and just enough humor to keep you hooked.
Today, we are taking a deep dive into two metabolic engines that literally define life on Earth.
We're talking about the process that fixes carbon into usable fuel and the corresponding pathway that generates the cellular reducing power required for all biosynthesis.
It's a really foundational piece of biochemistry. We're focusing on chapter 20 of our material, the Calvin cycle and the pentose phosphate pathway. And when you think about the scale, this is where molecular structure dictates global ecology.
Okay, let's unpack that a bit. Everything we eat, everything that fuels our bodies and builds our cells, it all fundamentally started as atmospheric CO2.
Exactly.
Photosynthesis is the gateway. It introduces all the carbon atoms into the living world. The process is so vast that, its signature is visible from space.
That's the perfect context. We often look at the Mauna Loa data, that long-term measurement of atmospheric carbon dioxide.
Right, the Keeling curve.
Exactly. And what you see is this relentless upward climb, but superimposed on that trend is a predictable, massive annual oscillation. It's that classic sawtooth pattern.
The planet breathing.
Precisely. That annual dip, which represents a colossal uptake of CO2, is overwhelmingly driven by the seasonal cycle of CO2 fixation. It's mainly the Calvin cycle in the massive terrestrial plant biomass of the Northern Hemisphere.
So it's a direct macroscopic visualization of this molecular pathway's global impact.
It is.
So our mission today is to get into the mechanics of those two interlinked cycles. Give us the overview. How does the Calvin cycle connect back to the light reactions we've talked about before?
Well, to set the stage, photosynthesis is traditionally broken down into two phases. You have the light reactions from chapter 19 that capture light energy and transform it into chemical energy.
Stored as ATP and
crucially, as biosynthetic reducing power, which is NADPH.
Okay, so that's the light reactions, and you have the dark reactions.
Right. The dark reactions, which include the Calvin-Benson cycle, take those high-energy products, the ATP and the NADPH, and use them to reduce fully oxidized carbon, CO2 gas, into a highly reduced form.
Hexose sugars.
Hexose sugars. These are the fundamental chemical fuels and building blocks for all life.
And this is where we find our central narrative for this deep dive, this idea of metabolic mirror images.
That linkage is absolutely key. If the Calvin cycle is the definition of reduction, taking oxidized carbon and using NADPH to make excesses, then the pentose phosphate pathway, or PPP, is its functional mirror.
So it's an oxidative pathway.
Exactly. It's oxidative. It takes hexoses, like glucose, and oxidizes them specifically to generate NADPH. They even share a number of enzymes and intermediates, which shows what our source material calls a beautiful evolutionary kinship.
Before we jump into the cycles themselves, let's just nail down that distinction between NADPH and NADH. Why does life need two separate currencies for transferring electrons? It seems redundant at first.
It's all about compartmentalization and dedicated function. Think of NADH as largely the currency of catabolism.
It's breakdown.
Right, the breakdown of molecules. NADH is primarily produced during glycolysis and the citric acid cycle, and its electrons are mostly destined for the respiratory chain.
Where they're used to generate a proton gradient, driving massive ATP synthesis, so it's an energy carrier.
Exactly. NADH is for power generation.
Got it.
NADPH, on the other hand, is the dedicated currency of analysis, the synthesis of complex molecules. When the cell is building things, fatty acids, cholesterol, amino acids, it needs electrons in bulk for all these reductive steps.
And NADPH provides that.
NADPH provides that reducing power, that little extra phosphoryl group attached to the ribose ring. It acts like a metabolic tag. It makes sure the cell can keep its general energy budget, the NADH-NAD plus pool, totally separate from its construction budget, the NADPH-NADP plus pool.
That clarity on the currency is essential. Now let's jump into part one, the Calvin cycle. This whole process happens in the chloroplast stroma, and our sources break it down into three main stages.
Yes, three really logical stages that ensure maximum efficiency. Stage one is fixation, where CO2 is captured and covalently attached to a 5-carbon acceptor molecule.
Okay, stage one, fixation.
Stage two is reduction. This is where the newly fixed carbon is turned into a 3-carbon sugar, which then becomes a hexose fuel.
And stage three?
Stage three is regeneration. You have to rebuild that 5-carbon acceptor molecule so the cycle can keep on fixing more carbon.
And stage one, it revolves around arguably the most famous and definitely the most abundant enzyme on the planet, rubisco.
The full name is ribulose 160 phosphate carboxylase oxygenase, a mouthful.
It is.
It's this massive enzyme, typically made of 8 large and 8 small subunits, and it catalyzes that crucial first step. The reaction begins when ribulose 1,4,and 5-bisphosphate, or RuBP, a 5-carbon sugar, is converted into a highly reactive anidylate intermediate.
So that anidylate intermediate is the key reactive species here.
That's right. CO2 then acts as an electrophile, condensing with the C2 of this anidylate.
Which forms an unstable 6-carbon compound.
Right, a beta-keto acid intermediate.
Yep.
And it is so unstable that it instantly hydrolyzes, which results in the production of 2 molecules of the 3-carbon compound, 3-phosphoglycerate, or 3-PGA.
So 1C5 plus 1C1 yields 2 C3 molecules, and our sources note this step is really, really thermodynamically favorable.
It is remarkably favorable. The reaction is highly exergonic with a delta G of negative 51.9 kilojoules per mole.
Wow.
So you might think, given its importance and how favorable it is, that rubisco would be a fast, efficient catalyst. But here is the central paradox.
It's not.
It is one of the slowest enzymes known.
This is the surprising fact about rubisco. Its maximal catalytic rate is only 3 zeros. You mean it only processes 3 molecules of CO2 per second?
That figure is just staggering when you compare to other enzymes that can turn over thousands of substrate molecules per second.
So how does it compensate for being so sluggish?
Sheer volume. Plants have developed a biochemical solution. Just make tons of it. Rubisco can account for up to 30% of the total soluble protein in a typical leaf. Its global abundance is a direct consequence of its microscopic inefficiency.
That inefficiency demands abundance. Now let's dig into what this sluggish giant even needs to become active. You mentioned it requires M-Gall binding.
It does. A divalent metal ion, usually magnesium, is absolutely essential for the mechanism. It helps stabilize the negative charges that develop when RuBP forms that anidylate intermediate.
Okay.
But the active site, where the magnesium binds, is only formed through a really complex regulatory step called carbamation.
And this involves a specific lysine residue.
You've highlighted the crucial distinction there. A non-substrate molecule is CO2.
So not the one being fixed.
Not the one being fixed, a different one. It reacts with the epsilon amino group of lysine 201 on the large subunit. This creates a negatively charged carbamate adduct. And only once that carbonate is formed can the magnesium ion bind securely.
And that creates the fully functional active site ready for the actual substrate, RuBP.
Precisely. So you need CO2 to be fixed, but you also need another CO2 molecule just to activate the enzyme that does the fixing.
That's amazing.
And in many plants, this process is so complex it requires a separate enzyme, rubisco activase, which actually burns ATP to pry off tightly bound inhibitory sugar phosphates from the active site so that the activating CO2 can get in and form that carbamate.
A brilliant way to make sure the process only runs when energy is plentiful.
Exactly.
Okay, so once rubisco has done its slow, vital work, stage 1 has produced 3-phosphoglycerate. Now we move to stage 2, reduction.
Stage 2 takes this oxidized carbon 3-PGA and converts it into a chemical fuel. It starts by consuming ATP to convert 3-PGA to 1,4,3-bisphosphoglycerate.
Which is a highly energetic intermediate.
Right. And it's then reduced to glyceraldehyde 3-phosphate, or GAP.
This whole sequence looks identical to the pathway in gluconeogenesis that runs in the cytoplasm. But the source notes a critical difference in the chloroplast.
The difference is the cofactor. In gluconeogenesis, the cytoplasmic enzyme uses NADH. But in the chloroplast stroma, the glyceraldehyde 3-phosphate dehydrogenase is specific for NADPH. This is the pathway where the cell uses that reducing power generated by the light reactions to convert the oxidized carbon into a reduced sugar. The result, GAP, is a 3-carbon molecule that can then be processed into fructose 6-phosphate, a 6-carbon sugar. And that completes the reduction phase.
Stage 3 is the hard part, right? It's the metabolic accounting needed to make sure the cycle doesn't run out of its starting material, rho-UBP. We have to take these C3 and C6 sugars and rearrange them back into a C5 sugar, the regeneration phase.
It's mathematically dense, but it's conceptually beautiful. We need to construct 3 C5 molecules, UBP, from 5 C3 molecules, GAP. This requires moving C2 and C3 fragments between different sugar backbones using a handful of specialized enzymes.
And this is where we first meet those 2 crucial transfer enzymes, which, spoiler alert, will reappear later in the pentose phosphate pathway.
Precisely. We use transketolase and aldolase. Transketolase, which needs the cofactor TPP, transfers a 2-carbon unit, and aldolase transfers a 3-carbon unit via an aldol condensation.
Okay, let's trace the flow step by step, because this is where the biochemical economy really shows itself.
Okay, so we start with fructose 6-phosphate, a C6, and GAP, a C3.
Right.
First, transketolase transfers a C2 unit from the F6P to the GAP. This gives you a 4-carbon aldose, erythrose 4-phosphate, and a 5-carbon ketose, niobulose 5-phosphate.
So C6 plus C3 becomes C4 plus C5?
Correct. Second, that the erythrose 4-phosphate combines with another C3 sugar, DHAP, via aldolase.
And that reaction forms a 7-carbon sugar.
C-dehydrogenase 1-color 7-bisphosphate, C7.
Got it.
Third, a chloroplast-specific phosphatase cleaves 1 phosphate off, giving you C-dehydrogenase 7-phosphate. Then fourth, we see transketolase again. It transfers a C2 unit from that C7 sugar to a third GAP molecule.
So C7 plus C3.
Becomes 2 C5 sugars, ribose 5-phosphate and another xylulose 5-phosphate.
That is a complex series of swaps. So through 4 steps involving those 2 key enzymes, we've successfully manufactured 3 different C5 sugars.
We have. And the final steps just convert these C5 isomers into the starting material, OUBP. The ribose 5-phosphate is converted by an isomerase. The xylulose 5-phosphate is converted by an epimerase, both giving you ribulose 5-phosphate.
And the cycle is completed when?
When phosphorubylgluconase phosphorylates it, consuming one final molecule of ATP to regenerate the rubP, the carbon acceptor.
That is the complete cycle. Let's step back and look at the immense energy cost required to run this engine 6 times to generate just 1 hexose molecule.
The stoichiometry is staggering. To incorporate 6 CO2 molecules, the cycle requires 18 molecules of ATP and 12 molecules of NADPH.
18 ATP and 12 NADPH, so that's 3 ATP and 2 NADPH for every single CO2 molecule fix.
It is a colossal investment of high-energy phosphate and reducing power. The light reactions have to be incredibly robust to support that kind of demand.
They absolutely do.
And this huge energy cost, it really defines the efficiency challenge for plants. Now, once the fixed carbon is successfully reduced to that hexose monophosphate pool, the plant has to decide what to do with it.
So now that we know the massive cost of production, let's look at the immense value of this output. What are the storage fates for this carbon fuel?
The plant has 2 primary options. First, starch. This is the long-term storage carbohydrate, a glucose polymer made right inside the chloroplast. It's less branched than our glycogen, but functionally very similar.
And what's interesting about starch synthesis is the activated precursor it uses.
Yes, that's a key detail. Unlike glycogen synthesis, which uses UDP glucose, starch synthesis relies on ADP glucose.
Why the different nucleotide?
Well, using ADP instead of UDP for activating glucose ensures that the synthesis pathway within the chloroplast is distinct and locally regulated. It separates it from any cytoplasmic synthesis pathways.
Okay, and the second major fate is sucrose, the transportable sugar.
Right, sucrose is synthesized outside the chloroplast, in the cytoplasm. The key challenge here is getting the fixed carbon out of the chloroplast. Hexosophosphates are tricky to transport across the membrane.
So what does it do?
So the 3 carbon intermediates, the triose phosphates like GAP, are exported via a specialized, highly abundant triose phosphate antiporter.
So GAP leaves the chloroplast and phosphate ions enter it, which maintains the internal phosphate concentration.
Precisely. Once in the cytoplasm, those C3 units are recombined to form F6P. That then combines with the glucose from UDP glucose to yield sucrose 6-phosphate. You remove the phosphate and you get sucrose, which is highly water-soluble and can be efficiently transported all over the plant through the phloem.
The entire Kelvin cycle is fueled by light. This brings us to part 2. How does the cell ensure this massive, costly cycle is only running when the sun is out? We need to talk about the elegant regulatory signals.
The regulation is tied directly to the physical consequences of the light reactions in the stroma. I mean, light doesn't just produce ATP and NADPH. It fundamentally shifts the physical and chemical environment where rubisco and the other Kelvin cycle enzymes live.
What are those light-driven environmental changes that, flip the switch?
As electrons flow during the light reactions, protons, H plus air, are pumped from the stroma into the thylakoid lumen. This massive proton shift causes 2 main things to happen in the stroma.
Great, what are they?
1, you get increased alkalinity. The stroma's pH rises from about 7 in the dark to 8 in the light. And 2, you get an increased magnesium concentration. As H plus ions flow out, Mu ions flow in from the lumen to maintain charge neutrality.
And those 2 simple changes, pH and MUR, directly coordinate rubisco activity.
They do. The increase in alkalinity, that pH of 8, strongly favors the necessary deprotonation of lysine 201, which promotes the formation of that carbamate adduct we talked about.
The step required to create the functional active site.
Right. And once that carbamate is formed, the now higher concentration of MgO is readily available to bind to it, fully assembling the active site and activating the enzyme. Light literally acts as a trigger for chemical activation.
That's one signal. What about a direct signal from the reducing power itself?
That's handled by the thioredoxin system. When light is present, reduced ferredoxin is plentiful. This reduced ferredoxin acts as the electron donor to an enzyme, ferredoxin thioredoxin reductase, which in turn reduces the protein thioredoxin.
And thioredoxin is a small protein with a key regulatory disulfide bond.
It is. So reduced thioredoxin is the central command.
Okay.
Absolutely. Reduced thioredoxin activates numerous key Calvin cycle enzymes, like the two bisphosphatases used in regeneration, fructose 1-furan-6 bisphosphatase and acetoheptulose 1-furan-7 bisphosphatase, by reducing and cleaving the regulatory disulfide bridges.
It chemically turns them on. Thanks, Beth.
And simultaneously, it uses this exact same mechanism to inactivate glycolytic or other degradative enzymes. This ensures the cell's entire metabolism is coordinated toward biosynthesis while the sun is out.
That is an elegant, multi-layered cascade. Light produces electrons, which activates a small regulatory protein, which then flips dozens of metabolic switches across the stroma.
And we also see NADPH acting as a direct signal. High NADPH levels can activate phosphorubylochanase and glyceraldehyde 3-phosphate dehydrogenase by promoting the dissociation of an inhibitory protein complex.
This regulatory coordination is so necessary because now we have to talk about rubisco's inherent flaw: photorespiration.
This is the enzyme's fundamental liability. We mentioned that the active site stabilizes a reactive NADPH intermediate. Well, although it's designed to react with CO2 occasionally, and especially when O2 concentrations are high and CO2 is low.
The active site mistakenly binds O2 instead.
Exactly. It functions as an oxygenase instead of a carboxylase. Under typical conditions, the oxygenase activity runs at about 1/4 the speed of the carboxylase activity.
And when it reacts with O2, what's the product?
It yields one molecule of the desired 3-phosphoglycerate, the C3, but also one molecule of a 2-carbon compound, phosphoglycolate.
And why is this phosphoglycolate so wasteful?
Because it's metabolically useless and it's toxic. The plant has to spend huge amounts of energy trying to recover that lost carbon in a really complex multi-compartment salvage pathway. It involves moving molecules across 3 different organelles: the chloroplast, the peroxisome, and the mitochondrion.
Okay, let's slow down and trace this necessary but painful salvage operation for the listener.
It starts in the chloroplast. The phosphoglycolate C2 is dephosphorylated to glycolate. Glycolate is then transported out of the chloroplast and into the peroxisome.
What happens in the peroxisome?
In the peroxisome, glycolate is oxidized to glyoxylate. This step consumes O2 and generates hydrogen peroxide, H2O2, which is a potent reactive oxygen species that has to be immediately neutralized by the peroxisome's high concentration of catalase.
Right.
Glyoxylate is then transaminated to the amino acid glycine. Glycine is still a 2-carbon molecule, and that's where the next organelle comes in.
Glycine travels into the mitochondrion. Why is that transfer necessary?
This is the core of the problem. 2 molecules of glycine have to enter the mitochondrion and react to form 1 molecule of the 3-carbon amino acid serine. And in this specific reaction, the conversion of 2 C2 units into 1 C3 unit 1 carbon atom is released as CO2.
Ah, so that's the definition of photorespiration. Consuming O2 in the peroxisome and releasing CO2 in the mitochondrion, all without generating any ATP or NADPH. It's just pure metabolic loss.
Exactly. We consume O2 to try and fix the error, but we still lose 1 carbon as CO2. The serine then returns to the peroxisome, where it's converted back to glycerate, and that glycerate finally moves back into the chloroplast, where it's phosphorylated, consuming another ATP, to re-enter the Calvin cycle as 3-phosphoglycerate.
So in this whole complex journey across 3 organelles, the cell spent energy, consumed O2, and lost 1 carbon just to recover 75% of the carbon that rubisco fixed incorrectly. Why did evolution settle for such a flawed enzyme?
While our source material suggests this is an inevitable failing tied to the geometry of the substrate, rubisco has to have a channel open for CO2 to enter, but O2 is also a linear molecule, and it's structurally simple enough to just slip into that active site and compete.
So it's a case of mistaken identity.
It's likely that rubisco evolved millions of years ago when the Earth's atmosphere was dominated by CO2 and had very low levels of O2. In that ancient environment, its lack of specificity wasn't a problem, so the evolutionary pressure to improve it was low.
But in the modern, oxygen-rich world, that failure becomes catastrophic, especially in hot, bright conditions.
That's the key environmental stressor. The oxygenase activity of rubisco increases far more rapidly with temperature than its carboxylase activity. So if a tropical plant gets too hot, its carbon fixation efficiency just plummets. And this intense selective pressure drove the evolution of some really complex workarounds.
This brings us to the famous C4 and CAM pathways, which are designed to create an internal CO2-rich environment for rubisco. Let's start with C4, used by plants like corn and sugarcane.
The C4 strategy relies on spatial separation. They divide the work between two distinct cell types: the mesophyll cells, which are exposed to atmospheric CO2, and the bundle-sheath cells, which hives rubisco and the full Calvin cycle.
So how does the mesophyll cell capture CO2 so efficiently?
It uses an enzyme called phosphenylpyruvate carboxylase, or PE carboxylase. This enzyme is far superior to rubisco for initial fixation because it is entirely specific for CO2.
Wow.
It cannot react with O2.
Ah,
It fixes CO2 onto a 3-carbon molecule, PEP, to form a 4-carbon compound, usually oxaloacetate, or its reduced form, malate.
so the mesophyll cell is the high-efficiency atmospheric capture unit.
That's precisely right. The C4 compound is then actively transported from the mesophyll cell into the protective bundle sheath cells.
Where it gets decarboxylated.
Exactly. Inside the bundle sheath, it's oxidatively decarboxylated, releasing a concentrated burst of CO2. This can raise the local CO2 concentration to levels 10 to 20 times higher than the atmosphere, effectively drowning rubisco in its preferred substrate.
By creating this high-concentration CO2 bubble, they nearly eliminate photorespiration. But we know this system comes at a premium energy cost. Let's trace that.
Right. To regenerate the starting material, the PEP, in the mesophyll cell, the 3-carbon pyruvate left behind has to be shuttled back and converted back to P. This conversion is catalyzed by the enzyme pyruvate pidekinase.
And this is where the cost calculation changes drastically.
It is. The pyruvate pidekinase reaction requires the equivalent of 2 ATP molecules to power this conversion and transport for every single CO2 molecule.
So wait, the standard C3 cycle already costs 3 ATP per CO2?
Correct. So if the C3 cycle costs 18 ATP per hexose and the C4 cycle adds another 2 ATP per CO2, that's 12 more ATP total. The total cost for C4 fixation is 30 ATP per hexose molecule.
30 ATP. That extra 12 ATP is the cost of actively pumping the CO2. It seems outrageously expensive.
It is, but in hot, bright environments where photorespiration in C3 plants might cause massive carbon losses, maybe 50% or more, the C4 plant is vastly more efficient overall. C3 plants dominate cooler zones where 18 ATP is enough, but C4 plants are the champions of the tropics.
Okay, now for the second adaptation, Crassulacean Acid Metabolism, or CAM, which uses temporal separation.
CAM is the adaptation used by arid-dwelling succulents, like cacti. Their primary survival concern is water loss. So to conserve moisture, they have to keep their stomata, the pores for CO2 absorption, closed during the hot, dry day.
Which means they can't photosynthesize during the day when the light is available.
So they fix carbon at night. When temperatures drop, they open their stomata and use that same highly efficient PPP carboxylase to fix CO2 into malate, which they store in large vacuoles.
They're storing the fixed carbon itself.
Exactly. When the sun rises, the stomata close. The stored malate is retrieved from the vacuole and decarboxylated internally, generating a huge concentration of CO2 right in the cell, which then feeds the Calvin cycle using the sunlight-generated ATP and NADPH.
So C4 spatially separates fixation and utilization, while CAM temporally separates them.
That's the core distinction. CAM is superior for water conservation, but because the storage capacity of the vacuole is finite, CAM plants are metabolically limited. They can't fix CO2 nearly as fast as C3 or C4 plants, which leads to their notoriously slow growth rates. It's the ultimate metabolic trade-off for survival in extreme drought.
We've covered the energy-consuming pathway that brings carbon into life. Now we make the great transition to part 3, the energy-generating pathway that gives the cell the reducing power it needs for synthesis and defense, the pentose phosphate pathway, or PPP.
The shift is fundamental. The Calvin cycle is unique to autotrophs, but the PPP is universal, occurring in the cytoplasm of virtually all organisms. Its dual purpose is critical, generating the vast supply of NADPH for reductive biosynthesis.
And providing the 5 carbon sugar precursors needed for nucleotide and nucleic acid synthesis.
Exactly. And like the Calvin cycle, the PPP has 2 distinct phases. The first is the oxidative phase, which is irreversible and generates the NADPH. The second is the non-oxidative phase, which is reversible and handles the interconversion of sugars to meet the cell's changing needs.
Okay, let's detail that oxidative phase, starting with the critical control point.
The first step is the dehydrogenation of glucose 6-phosphate, or G6P, to 6-phosphoglucono-delta-lactone. This reaction is catalyzed by glucose 6-phosphate dehydrogenase, or G6PD, and this is the rate-limiting control point for the entire oxidative branch.
And this enzyme's cofactor specificity is essential to its function, right?
It is absolutely essential. G6PD is highly specific for NADPO. If you look at its affinity, its Chiari value for NAD is about 1,000 times greater than its Chiari for NADPO.
1,000 times.
This extreme preference ensures the reaction is dedicated solely to generating the biosynthetic currency, NADPH, not NADH. This step generates the first molecule of NADPH.
What follows the lactone formation?
The lactone is quickly hydrolyzed to 6-phosphogluconate. Then a second oxidation occurs. 6-phosphogluconate dehydrogenase catalyzes an oxidative decarboxylation, releasing one molecule of CO2 and yielding the 5-carbon ketose, ribulose 5-phosphate.
And the second step generates the second molecule of NADPH.
It does.
So one molecule of G6P entering the oxidative phase yields 2 NADPH, 1 CO2, and 1 C5 sugar. Now, for the non-oxidative phase, if the cell doesn't need that C5 sugar, this phase is all about recycling it back toward glycolysis.
That's its primary purpose, flexibility. The ribulose 5-phosphate must first be isomerized to ribose 5-phosphate, the precursor for nucleotides, or epimerized to xylulose 5-phosphate. Then the goal is to convert these C5 molecules back into the C6 sugar fructose 6-phosphate and the C3 sugar glyceraldehyde 3-phosphate.
And we are reunited with the two critical transfer enzymes we first saw running the regeneration phase of the Calvin cycle, transketolase and transaldolase.
We are. This is a remarkable demonstration of evolutionary economy. The same enzymatic machinery is used for a reduction in the Calvin cycle and for oxidation and recycling here in the PPP. But this time, let's focus on the brilliant, yet different, chemical mechanisms they use.
Let's start with transketolase, which transfers a C2 unit. We know it requires thiamine pyrophosphate, TPP, as a prosthetic group. How does TPP actually enable this CC bond transfer?
TPP is the star of this show. The core of its activity is in the thiazole ring, specifically the C2 carbon, which is naturally acidic. When it's ionized, this carbon forms a powerful nucleophilic carbanion.
Okay.
This carbanion attacks the carbonyl group of the ketose donor substrate, like gazellose 5-phosphate.
So that attack releases the first aldose product, leaving a 2-carbon fragment attached to the TPP.
Exactly. This 2-carbon fragment, a glycolaldehyde unit, is temporarily bound to TPP. And the key to stabilizing this highly reactive fragment is the positively charged nitrogen atom within the thiazole ring.
It acts as an electron sink.
A powerful electron sink. It stabilizes the negative charge on the attached 2-carbon unit through resonance. This activated unit then attacks the carbonyl group of the new aldose acceptor, forming a new CC bond, creating a new ketose product, and freeing the TPP to start again.
That structural feature, the charged nitrogen stabilizing the intermediate, is the crucial piece of the puzzle for transketolase. Now, contrast that with transaldolase, which transfers a 3-carbon unit, but does not use a prosthetic group.
Transaldolase uses an equally elegant but entirely different mechanism. Instead of a prosthetic group, it uses a covalent intermediate formed with one of its own active site residues.
A lysine residue.
A covalent intermediate, how does that form?
The carbonyl group of the ketose substrate forms a covalent bond with the epsilon amino group of the active site lysine. This bond is known as a Schiff base, or a protonated amina.
So the lysine itself becomes the carrier. It does. And once that Schiff base is formed, the enzyme cleaves the bond between C3 and C4 of the substrate, transferring a 3-carbon dihydroxyacetone unit to the lysine. The resulting fragment is a carbanion. But unlike with TPP, here, the protonated Schiff base acts as the electron sink.
Its positive charge stabilizes the negative charge on the carbanion.
Right, through resonance. It allows that C3 unit to be held just long enough to be transferred to the acceptor aldose molecule.
That is a phenomenal comparison. Both enzymes achieve the same goal, creating a CC bond through a stabilized carbanion intermediate, but one uses a permanent cofactor, TPP, and the other uses a temporary protein-derived covalent intermediate, the Schiff base. It's engineering efficiency in action. This incredible metabolic flexibility brings us to part 4, coordination, regulation, and clinical relevance. Since G6P sits at this central metabolic junction, it can go to glycolysis for ATP or the PPP for NADPH. How does the cell decide which pathway to favor?
The decision is based purely on the cell's current demand for NADPH. We already identified the rate-limiting step as the G6PD reaction. Well, that reaction is rigorously controlled by the concentration of the cytoplasmic electron acceptor, NADP.
So if the cell is well supplied with reducing power high NADPH, what happens?
If the NADPH and ADPH is a high, meaning NADPH is abundant, the low concentration of NADPH slows the G6PD reaction simply because the electron acceptor is scarce.
Supply and demand.
Exactly. And what's more, the product, NADPH, is a potent competitive inhibitor of the enzyme. It physically blocks the binding site for NADPH. This tight control ensures the cell doesn't waste energy generating and reducing power it doesn't immediately need.
This regulatory switch allows G6P to be metabolized in 4 distinct modes, perfectly matching supply to the cell's dynamic demands. Let's walk through them.
Okay, mode 1 is when the cell needs ribose 5-phosphate, much more than it needs NADPH. Think of rapidly dividing cells, like in bone marrow or tumors, which are synthesizing DNA at an accelerated rate and need tons of nucleotide precursors.
So how does the cell maximize R5P without generating NADPH?
The oxidative phase is mostly suppressed. G6P enters glycolysis to generate fructose 6-phosphate and glyceraldehyde 3-phosphate. Then, the non-oxidative enzymes of the PPP run in reverse.
In reverse.
In reverse. They use the F6P and GAP to synthesize R5P. This shunting provides high precursor supply with minimal NADPH generation.
Running the pathway backwards? That's a brilliant adaptation. Okay, mode 2 sounds like the steady-state default.
It is. It's the simplest. When the cell requires both NADPH and R5P in roughly equal measure, G6P just runs through the oxidative phase, yielding 2 NADPH and 1 R5P directly.
Now for mode 3, maximum NADPH generation. This would be for, say, liver and adipose tissue where massive amounts of reducing power are needed for fatty acid and cholesterol synthesis.
This is where the recycling is maximized. The goal is to completely oxidize G6P to CO2 using the PPP. It's a 3-step process.
What is step?
1. G6P enters the oxidative phase, producing NADPH and C5 sugars. 2. The non-oxidative enzymes convert those C5 sugars into F6P and GAP. And 3. Crucially, these C6 and C3 glycolytic intermediates are immediately fed back into the gluconeogenic pathway to regenerate G6P.
So the C5 sugars are effectively converted back into G6P, which can then re-enter the PPP oxidative phase again and again. It's a loop.
That's the loop. For every 6 molecules of G6P that enter this recycling process, 5 are successfully recycled, while the equivalent of 1 G6P is completely oxidized to 6 CO2 molecules.
And the net equation for that loop?
G6P plus 12 NADP goes to 6 CO2 plus 12 NADPH. This maximized recycling achieves the highest possible yield of reductive power.
12 NADPH per oxidized hexose. That's how the liver powers its detoxification and synthesis. Okay, finally, mode 4. Balancing the needs for both NADPH and ATP.
In mode 4, G6P produces NADPH via the oxidative phase. The resulting C5 sugars are then converted to F6P and GAP, but instead of being recycled back, they feed forward directly into glycolysis. This results in pyruvate, which can then be oxidized further to yield maximum ATP and NADH.
This 4-mode system really shows how central G6P is and how small changes in the NADP concentration can fundamentally redirect carbon flow. You mentioned this metabolic shunting is often a hallmark of cancer cell metabolism.
It is. Cancer cells often exhibit the Warburg effect, but they still need massive amounts of building blocks. They often coordinate modes 1 and 3 by shunting glycolytic intermediates into the non-oxidative PPP for R5P for nucleotides and into the full recycling loop, mode 3, for NADPH, which they need for the high rate of fatty acid synthesis for new membranes.
And targeting the shunting mechanism is a major avenue for cancer research.
It is.
Now let's pivot to the protective role of NADPH, particularly in defending the cell against reactive oxygen species, or ROS.
The generation of NADPH by the PPP is the cell's frontline defense against oxidative stress, things like peroxides generated by normal metabolism. The primary detoxifying agent is the tripeptide glutathione, or GSH.
Glutathione is the antioxidant that sacrifices itself.
Exactly. The enzyme glutathione peroxidase uses 2 molecules of GSH to reduce a harmful peroxide to harmless alcohol or water. And in the process, GSH is oxidized to its disulfide form, GSSG.
And once it's oxidized, it's useless for defense until it's regenerated.
Correct, and this is where NADPH comes in. The enzyme glutathione reductase utilizes the electrons carried by NADPH and only NADPH to reduce GSSG back to 2 molecules of active reduced GSH. This ensures the cell's peroxide defense shield is constantly recharged.
So if you lose the ability to generate NADPH, you lose the ability to regenerate that GSH defense system.
And that failure chain brings us to the clinical highlight, G6PD deficiency. This malfunction of the rate-limiting enzyme in the PTP is the most common enzyme deficiency worldwide, affecting over 400 million people.
And why is the pathology most severe in red blood cells?
This is a classic case of metabolic dependency. Mature red blood cells lack mitochondria. They have no oxidative phosphorylation, no citric acid cycle. They're almost entirely reliant on glycolysis for ATP and entirely reliant on the PPP and specifically G6PD to generate NADPH.
Other cells can compensate, but not red blood cells.
Not in the same way now.
So what triggers the acute crisis in a person with G6PD deficiency?
The crisis is triggered by exposure to oxidative stressors that overwhelm their limited GSH supply. These can be specific antimalarial drugs like primacaine or chemical agents in certain foods, most famously the purine glycoside vicine found in fava beans. That's the condition known as favism.
When that oxidative agent hits, what is the immediate consequence inside the red blood cell?
The existing GSH is rapidly oxidized to GSSG, but because G6PD is deficient, there's no NADPH to regenerate it.
Yeah.
Oxidative stress skyrockets. The ROS start to damage the cell's components, particularly hemoglobin.
What happens to the hemoglobin?
It aggregates and denatures, forming these dense, insoluble clumps called Heinz bodies that attach to the cell membrane. This physical damage compromises the cell structure, leading to massive acute rupture, hemolytic anemia.
It's a debilitating disease, yet the genetic trait is widespread in specific geographic regions. This brings us back to the evolutionary paradox.
The prevalence of G6PD deficiency is highest in regions endemic to malaria, and it offers a profound insight into evolutionary trade-offs. The deficiency confers significant protection against the parasitic killer, Plasmodium falciparum.
If the cell is compromised, how does that protect it?
The malaria parasite needs a high level of NADPH for its own growth and replication inside the red blood cell. By compromising the PPP, the deficient cell creates a highly oxidized, low NADPH environment.
So it starves the parasite.
It starves the parasite of reducing power. And what's more, the infection itself increases oxidative stress. The compromised cell can't cope and is often destroyed quickly, taking the newly infected, still developing parasite with it. The parasite never reaches maturity and can't spread the infection.
So the genetic defect offers a survival advantage against a major parasitic threat, even if it carries the risk of severe anemia when triggered. It's an elegant, if brutal, natural optimization.
So to summarize our deep dive into Chapter 20, I think the core takeaway is the masterful economy of molecular life. The Calvin cycle is the definition of reduction, using ATP and NADPH to fix carbon. The pentose phosphate pathway is its functional mirror, an oxidative pathway that takes reduced carbon and generates the NADPH and precursors needed for everything else.
Right.
And the fact that these two pathways, serving such disparate goals, global carbon fixation versus internal cellular defense share complex enzymatic machinery like transketolase and transaldolase, just shows the brilliant efficiency of evolution in reusing tools for different jobs.
And here's where it gets really interesting for me. When you look at the entire scope of life, from the global scale of carbon cycling to the microscopic scale of a single red blood cell's defense, you see that biological systems are always making these highly constrained trade-offs. Rubisco's ancient, imperfect structure forced environmental adaptations like C4 and CAM, demanding a significantly higher ATP cost just to capture carbon efficiently in specific environments. And at the same time, a genetic flaw like G6PD deficiency, which risks deadly hemolytic anemia, evolved as a potent defense mechanism against malaria.
It's incredible.
It shows that the chemistry of survival is never truly about perfect efficiency. It's about constant optimization against immediate environmental threats.
Well said.
Thank you for joining us on this deep dive into the cycles that power the living world. We hope this gave you a few new aha moments about the necessary imperfections and brilliant solutions found within biochemistry. Until next time, keep digging into the details.